Chapter 4. In this chapter, we're going to be talking all about living chemistry, the chemistry that we need to fully understand how cells work and how bacteria live and grow in our environments. The learning outcomes for this chapter, by the end of the unit, are to correlate the structure of a macromolecule with its function in the cell. This is a huge theme in biology, is that the chemical structure of something in a cell or in a molecule really helps it perform its function. And so I'd like you guys to be able to think about, for each of the types of molecules we talk about, how is the structure set up in a way that helps that molecule do its function. Second, I'd like you to predict the effects of temperature, pH, and solute concentration on cells. This can really be important when we talk about disinfectants, or we talk about how do we prevent infection, or how do cells survive in different environments. We really need to understand this concept. And then last, describe how host cells can utilize membranes for defense and how some pathogens can subvert or get past these mechanisms. So we'll be talking all about those things here in this chapter. First, let's start with our opening case study. In 2010, there were heavy rains in Pakistan, sort of an unusually high and long rainy season. And our case is about a 10-year-old boy from Pakistan who had profuse diarrhea and extreme thirst. And his diarrhea was so intense, there was gallons and gallons of diarrhea every day. And you could see flecks of white material, what they call sort of rice water stools. And that white material is parts of the intestinal lining. Because of the rain and the lack of clean water, there is always a high rate of cholera present, and sure enough, when the nurse examined the stool, they found through dark field microscopy, they found these very motile, so these bacteria that were moving around. They were comma-shaped rods or comma-shaped bacilli, and that's very characteristic of the bacteria Vibrio cholerae. And so here is a scanning EM picture of that. You can see it's somewhat of a curved rod shape, and they have these single, really nicely arranged flagella that help them move around. So under the microscope in a dark field preparation, they move around a lot and look like little sparkly stars. Now, why are we talking about cholera at the beginning of our chemistry chapter? Well, cholera produces a toxin, and you can see that toxin sort of landing here. It's this AB5 toxin is what we call it, and it binds to the surface of a epithelial cell in the gastrointestinal lining. So this is a part of the intestine, and it binds to a particular sugar on the outside surface of the cell. It gets taken in through a process called endocytosis, which we'll learn about later in this chapter. And basically what happens is through a series of reactions, it causes massive amounts of chlorine ions to be excreted. Along with that come the positive ions, potassium and sodium, and water to help balance that out. So you can see this toxin, because of its binding, the fluids inside the cell and inside the tissues are all being expelled, and that's where the diarrhea comes from. Cholera is 50% fatal if left untreated, and the most effective treatment for that, you can see on this previous slide here, are these oral rehydration salts. So again, adding back in those important salts and ions along with some glucose can help prevent the massive dehydration and reestablish the ion balance on the inside and outside of the intestinal lining and prevent the massive diarrhea from occurring. So that chemistry of the intestinal lining becomes very important, and that's why we talk about that case in this opening chapter. Now, in section 4.1, it goes through a lot of really basic chemistry review that you should have had as part of your prereq. So I'm going to very quickly just touch on a few things. There's lots of different ways that we write molecules. So we have these electron orbital diagrams. We have these drawings here. You can see a common molecule we'll encounter is methane, CH4, and water. The major elements that we see in biology really include just these top 7 molecules, right? So hydrogen, carbon, nitrogen, oxygen, sulfur, phosphorus, and sodium. Beyond that, the biologists don't necessarily care much about the other types of elements. Here's some more: chlorine, potassium, calcium, magnesium, and iron that all become important in living cells. Okay, so we know that different atoms can lose an electron and become positively charged, or gain an electron and become negatively charged. So those are those ions we were just talking about in cholera. Those salts can be really important. So here we see the sodium ions being attracted to the chlorine ions with an ionic bond between oppositely charged atoms, we'll see some examples of ionic bonds found within proteins and other things, including salts. And then water. Water is really important in biology. Most of the cell is made up of water, and water has some pretty unique properties. So this is an amphipathic molecule, meaning it has a partial negative charge and partial positive charge. So we see these two different partial charges on this molecule that allows it to create these hydrogen bonds, right? So we have this polar covalent bond between the hydrogen and the oxygen, but the oxygen is more electronegative, so it's kind of a bully. It steals the electrons more of the time. Now that means that these partially negatively charged oxygens are attracted to the partial positive charges, and that's what we call a hydrogen bond. The hydrogen bonding between water molecules is essentially why we can have biological life on this planet. It keeps ice from sinking, so ice floats. That's pretty amazing for how life formed on this planet and how we can continue to have ocean life. It also means that those extra hydrogen bonds make water very stable, so it's hard to boil it and create steam, and it's hard to break those things up. The partial charges interact with lots of other molecules, so you'll see that in the case of salts, the ions can be sort of neutralized by the partial charges of the water that surrounds them. So we have the positive ion and all the oxygens facing it, and the negative ion here with all the positive hydrogens facing it. Now, nonpolar molecules, when put into water, tend to aggregate together. So if you drop a bunch of oil drops into a glass of water, they will congregate together in this nonpolar hydrophobic area, and the water will sort of just arrange around the outside. So water is hydrophilic. Obviously, it's self-loving, right? It likes all those charges and those interactions, and nonpolar or hydrophobic things tend to cluster. We'll talk about this property as being really important in the organization of membranes. Okay, so that's our sort of review of 4.1. If there's anything that you want to go back in and look in more in depth, please do so. Now, some common features of organic molecules. We're going to be talking primarily about the big organic macromolecules in this chapter. That's where we're going to spend most of our time. And we know that some common features of those are that they contain these carbon-carbon bonds. They tend to be large or have lots of subunits. We'll see examples of every type of macromolecule having a monomer subunit that gets put together with larger complexes to make these giant polymers. And they tend to have some of the same kinds of functional groups, and that's what's shown in our table here. So we'll see parts of these organic molecules having alkanes, amino groups, carboxyl groups, esters, hydroxyls, and carbonyls. We'll be talking about four major classes of macromolecules. The first one here is the lipids. So lipids are made up of fatty acids as their monomer, and those fatty acids basically contain sort of a hydrocarbon chain, so carbons and hydrogens that are bonded together. So lipids, the main function of a lipid is to store energy in the cell. And they do that because they have high numbers of these carbon-hydrogen bonds. Those bonds are really stable, they're non-polar bonds, and they release a bunch of energy when they're broken. So that's what makes lipids a really great storage of energy. We can see that these fatty acids come in two distinct sort of flavors. If all of the hydrocarbons have a single bond, we have filled in with saturated with hydrogens, we call this a saturated fatty acid. If there are some double bonds within this hydrocarbon chain, it's not saturated with hydrogens. We could add more hydrogens. So we call this an unsaturated fatty acid. And you'll notice that it has sort of a kinked structure. It's not a straight line. When we talk about the role of lipids in membranes, we'll talk about how the arrangement of saturated fatty acids and unsaturated fatty acids can actually help make a membrane more or less fluid. So the properties of these lipids become really biologically important. When we put those monomers, those fatty acids together, we can make a couple of different types of molecules or polymers. One of the things, if we have 3 fatty acid molecules and an alcohol group called glycerol, we can make triglycerides. Triglycerides are also commonly thought of as sort of fat molecules, and these are high energy molecules that get stored. We see this in animal tissues as a way to insulate and regulate body temperature, but also as, again, a source of energy. We'll see later in the chapter that in addition to triglycerides, if you replace one of these fatty acids with a phosphate group, you can get a phospholipid. And we'll talk about the purpose and the structure and arrangement of phospholipids in section 4.5. Okay, so the next category of macromolecule that we want to talk about are carbohydrates. So carbohydrates have a monomer called a monosaccharide, and these are carbon-based molecules that have between 5 and 7 carbons. So here's our sort of classic 6-carbon sugar, a hexose. We typically number the carbons based on the carbonyl group, right? So this is glucose. And then if we move the carbonyl group to the second carbon, right, we call that a fructose. So these 6-carbon sugars can come in lots of different flavors depending on where that carbonyl group might be, but they all tend to have the sort of same kinds of functions. So the function of carbohydrates are in energy, so they also store a lot of energy. We see a lot of those carbon-hydrogen bonds. They tend to be nonpolar or non-reactive. They're very stable and inert. And we also see carbohydrates participating in a lot of cellular structures. Now they don't make those cellular structures as this monosaccharide or building block. What we see is that these carbon structures, these monosaccharides, can loop around and form these ring structures. So here we see the ring structure of glucose, alpha glucose and beta glucose. Those ring structures then can come together and form bonds and create a disaccharide, 2 monosaccharides together. Now when those come together with other monosaccharides, we can get very, very long carbon chains. So these links, these circular carbohydrates can form very, very long polymers called polysaccharides. And that's where we start to see them playing a role as cellular structures. These big, large sugar molecules that can play a role in a number of different things. They're part of the cell wall. They can be the capsules made out of carbohydrates. They can serve as signaling and adhesion molecules. Okay, so the next macromolecule that we want to talk about are the nucleic acids. So nucleic acids are made up of a monomer consisting of nucleotides. So nucleotides, so this nucleobase that we see here, come in basically 5 flavors. So there's a nitrogen ring, nitrogen carbon ring here. So we see adenine has the 2 rings together. Guanine also has these 2 rings together. So we call those purines. And then we have 3 nucleotides that don't have double rings. They just have a single ring, but they still contain nitrogen. So we have cytosine, thymine, and uracil. Now that nucleotide base right here in the purple is attached to a sugar and a phosphate group. So this 5-carbon sugar is called ribose, and in some nucleic acids we can see the difference of this oxygen makes it deoxyribose. So we have ribose and deoxyribose as this 5-carbon sugar, and then lastly we have a phosphate group. It's important to remember this phosphate group. We'll see in a minute how you can have more than one phosphate group attached to these nucleotides. But those phosphate groups are negatively charged. And one of the principles of chemistry we know is that, in all of science, is that like charges repel each other. So having more than one of these negative phosphates in the same molecule can store a lot of energy because those phosphate molecules want to split apart. It's very energetically favorable. So here's that example. So we have here the nucleotide base that has adenine, so we have double rings here, and it's attached to this 5-carbon sugar ribose, okay? And if we have one phosphate group, we call it an adenosine monophosphate, so mono meaning 1, AMP. If we have 2, we have adenosine diphosphate, or ADP, and then if we have 3, we have triphosphate. So ATP with 3 phosphates, 3 of these negative charges, 4 if you really count this one on the end, all smashed together is an incredibly high energy molecule. I always think about the springy snake that you smash into the can, right? So that there's a lot of energy stored in there. That snake does not want to be smashed into the can. And so in ATP, when we have all 3 phosphate groups, this molecule is storing a lot of energy because these negative charges want to split off. So we'll see ATP used as a high energy molecule when we talk about more about metabolism in chapter 6. Okay, so nucleic acids come in 2 different sort of flavors. We have DNA, deoxyribonucleic acid, and RNA. Let's spend a second looking at the structure of DNA. So DNA, again, has this deoxyribose, and it has these phosphate groups. So that's this blue thing here in the phosphate group here. And we've talked about these nucleotides. Now notice how the nucleotides come together in unique ways and make hydrogen bonds. So here we have adenine on the left, one of the purines, binding to the pyrimidine thymine. And as they are arranged, you can see there are two hydrogen bonds between the A and the T. Now down here we see we have cytosine, the pyrimidine, and we have guanine as a purine. And together these two nucleotides make three hydrogen bonds in a unique combination. So we call this base pairing, where A nucleotide bases pair with T nucleotide bases. So A pairs with T, and then C pairs with G. And because of their chemical structure, that's really the only way that those four types of nucleotides can come together, C's and G's with A's and T's. And so we'll use that to really understand how the structure of DNA lends itself to its function, because if you take away half of a nucleotide, a DNA molecule, you can recreate the other half because of this nucleotide base pairing. So we talk about DNA as being double-stranded because it has two sugar phosphate backbones and the nucleotides are base paired together. So on this side we see sugars and phosphates with nucleotides, and on this side we also have a sugar phosphate backbone with the nucleotides. Now, one thing to point out, we'll talk more about this in genetics, is that here we have the phosphate group on the 5' end, and we have the oxygen on the 3' end. So we talk about the carbons, we label the carbons, and this one's attached to the 5' carbon. So we talk about this as the 5' end of the DNA, and over here, this is oxygen that's attached to the 3' carbon, so we call this the 3' end. Notice that the phosphate 5' end is opposite the 3' end. So the arrows here are what we call anti-parallel. They go in opposite directions. We'll talk more about why that is in later chapters. Now, if you sort of cartoonize that a little bit more, you can see here again we have our base pairing rules, C's and G's. We have this purple ribbon is representing our sugar phosphate backbone. And you can see that if you arrange these things together, they twist into a double helix. Here's a space-filling model of that. You can see all the different molecules coming together. And you'll see that there's sort of this characteristic major groove and minor groove, right, that happens in the DNA. We'll again talk more about that, but when we think about the function of the DNA, this is an energy or sort of an information storage molecule. So when we think about the code or the recipe that makes a cell, it comes from this cookbook, right? So the DNA molecule is like a large cookbook filled with recipes, and each recipe is encoded in this language of A's, T's, C's, and G's. Now this cookbook is pretty great because you can split it in half and give half to a daughter cell or an offspring cell. And we can do that because the structure of DNA is double-stranded. So if I take away one of these strands, I can build it again because of those unique base pairing rules. So DNA is our storage of information. It's very stable. It's very inert. It doesn't have a lot of reactivity, and it contains all of that code, and it can be split in half. So that's how the structure of DNA is really related to its function. Now, the other kind of nucleic acid is RNA. So RNA has a single sugar phosphate backbone. The sugar in RNA is ribose instead of deoxyribose. And you can see here that we have this strand of RNA. We can see the bases, adenine, cytosine, cytosine, adenine. But as you go along, you won't see any thymine or T. Instead, you'll see uracil. So uracil is the base that is used in RNA, whereas thymine or T is used in DNA. So it uses slightly different bases. Now RNA, even though it's single-stranded, it can still form a number of different kinds of structures. So here's an example of a transfer RNA where the RNA itself has wrapped back around itself. So here we have a 3' end and here we have the 5' end. But as the RNA wraps around itself, it makes some of those base pairs that we see between the different nucleotides. So RNA has a large diversity in structure because of its single-stranded nature, and it can be both a functional molecule, meaning it can serve as sort of like a part of an enzyme or act like an enzyme, or it can simply transfer that coded information within the A's, C's, G's, and U's from DNA into protein. So we'll talk more about that process in our genetics chapter. Okay, Our last macromolecule that we need to talk about are proteins. So proteins, the monomer for proteins, are amino acids. And here we see sort of a diagram of a single kind of amino acid. So we have an alpha or central carbon. On one side we have a carboxyl group, and the other we have an amino group. So carboxylic acid helps us name these amino acids. And then notice we have this R up here. This indicates a side chain. So with DNA and RNA, there are basically 4 different bases in DNA and 4 different bases in RNA. That's the code is made out of those 4 letters. In proteins, we have up to 20 different amino acids that are commonly used. So in each one of these amino acids, we see that central carbon, and then in yellow is that R group. And so we see lots of different combinations of kinds of chemicals found within that R group. As we start to put these together, you can hopefully already appreciate that proteins are going to be a little bit more complex. So we have groups of charged amino acids, we have groups of polar amino acids, hydrophobic amino acids, cyclic amino acids that have sulfur that can do bonding with their sulfur. We have very special other kinds of amino acids that have other properties. So these amino acids have a lot of diversity and can give lots of different kinds of functions to proteins. So proteins are much more complex. We'll talk about how they have 4 different levels of structure, and they do so many different jobs in the cell. So the DNA is the instructions to make these proteins. We have sort of a separate way of storing the information on how to make them, but once we make them, they can do everything that the cell needs and requires. So enzymes are made out of proteins, parts of the cell like the cell wall, the flagella, the cytoskeleton, the proteins that are wrapped around the DNA, all of those things are made out of protein. Lots of signaling molecules are made out of protein. Lots of our cellular and animal structures are made out of protein. So proteins can be very, very diverse in their function. Now, how do we get to those diverse functions? We have a very complicated structure. So again, this comes back to that structure-function relationship. So a complicated structure gives a very complicated function. So when we have 2 amino acids coming together, we use a condensation reaction to make water, which is very favorable, and we call that a peptide bond. So we take out this water, and now we have 2 amino acids that are attached together. So here we have R group 1 and R group 2. So this is called the primary structure of the protein. It is simply the different amino acids all attached together through peptide bonds. And here we see an example. Here we have serine, phenylalanine, aspartic acid, methionine, and alanine. In proteins, instead of a 5' and 3' tail like in DNA, we have an amino terminus where the amino group is, and a carboxyl terminus or C terminus where the carboxylic acid group is. Now, primary structure, like I said, is just those amino acids attached in a line. Here we see that sort of up close. Okay, so secondary structure is formed when hydrogen bonds within the amino groups and the carboxyl groups come together, and they can come together in sort of 2 basic conformations. We have this alpha helix where the amino acids are sort of arranged in a spiral or helical shape, and you can see the hydrogen bonds that hold that in place in red. We also have something called a beta pleated sheet. So the amino acids can fold in such a way that they sort of make a flat structure that sort of twists back and forth. So secondary structure, again, has nothing to do with the R groups. It is simply a way to fold those carboxyl and amino groups in such a way that they make hydrogen bonds. So here we see some different kinds of ribbon diagrams where you can see those red alpha helices and those blue beta pleated sheets. Okay, now tertiary structure is when we start to see the interactions of the different R groups. So here we can see R groups that have hydrophobic interactions coming together on the inside. Amino acids that have sulfurs like to form disulfide bonds. We have amino acids with different charges, so negatively charged and positively charged will come together to make ionic bonds. So depending on the order of the different amino acids, you'll see hydrogen bonds, hydrophobic interactions, covalent bonds, and ionic bonds forming between that. And that can give you an incredible diversity in structure of the protein. So tertiary structure is where we really start to see this 3-dimensional protein being developed. Some proteins stop at tertiary structure, so they simply use those R group interactions to create their final structure. Others, like this particular, this is an antibody, use a 4th level of structure called quaternary structure. Quaternary structure is when more than one protein come together to make a larger complex. So here you see in green one polypeptide chain, and in yellow another polypeptide chain. So here two different tertiary structures come together to make a complete molecule like this immunoglobulin or antibody molecule. Another example that has quaternary structure is hemoglobin inside of our red blood cells. Okay. So when we talk about energy and how do these chemical reactions occur in the cell, we need to remember that every time we have energy and we transform it in some way, we get some usable energy, but we also lose a little bit of that energy. So every energy transformation has a little bit of waste. And so the cell needs to be incredibly careful about how it manages its energy in the cell. And one ways that we do that is we use enzymes. So enzymes are made out of protein. We learned a minute ago that those proteins can have incredibly specific structures because of all 20 different amino acids. So many of the jobs that proteins have in the cell are these enzymes. Here you can see that the energy it takes to do a reaction is in blue. So if you just have the reaction and you need to do it, you have to drive up the energy for that energy to go from reactant to product. So we have to add some energy to make the reaction go in blue. And that energy is pretty high. And we get a free energy release that's the same, sort of a negative free energy release. The energy goes forward. Now, if we add an enzyme, so this is the orange line, we can reduce that energy of activation, make the reaction happen better, and we still get all of this delta G, right? So we still get the same sort of release of energy, but with much less input. So the reaction rate can happen faster. So when energy is really tight, we simply rely on these enzymes to catalyze all of our chemical reactions within the cell and make sure that they happen. Now that means that those enzymes, we have to keep them very happy. And all of those different kinds of chemical bonds we talked about in the tertiary and secondary structure can be disrupted. They can be disrupted through temperature and pH changes. So temperature, when the molecules aren't moving very quickly, it's very hard to have the reactions occur. So in low temperatures, the molecules move slowly and the energy and enzymes move slowly as well. When you start to increase the temperature, those molecules move faster and the enzymes move faster, and we see a high rate. But every enzyme has an optimum temperature, and then beyond that temperature, those bonds begin to break. So the hydrogen bonds begin to fall apart, those interactions between the R groups of the amino acids begin to fall apart, and the protein goes from its quaternary or tertiary structure back to its primary structure in a process we call denaturation, when the protein basically falls apart. And so higher than optimal temperatures can cause that to happen. Now pH is something that we also talk about. So here we see the pH scale from 0 to 14. We know that pure water is sort of the happy place, that's pH 7, and any deviation from that, then we get either a harmful or sort of a reactive condition, all the way from liquid drain cleaner to battery acid, right? So pHs, the disruption in the balance of hydrogen ions, can break apart all of those hydrogen bonds within a protein. So greater than or less than the optimal pH for an enzyme, those proteins will fall apart and denature as well. All right. So let's talk now about membranes. So when we think about membranes, there's a lot of words that we use. So we'll use, your book uses the term cellular membrane or cell membrane, but we also know that those can be called cytoplasmic membranes or simply plasma membranes in other textbooks. So I want to acknowledge that all of those words mean the same thing. It means the division of the inside of the cell from the outside of the cell. When we look at this diagram here, we can see sort of the outside of the cell, we have the extracellular fluid, we have the receptors or things that are attached to the outside of the cell that might interact with the outside environment. We have this phospholipid bilayer that we'll sort of zoom in and look at a little bit on in the next slide. And then we have the cytoplasm. This is the inside of the cell. So here we're talking about a membrane, a lipid structure, that separates the outside from the inside of the cell. Now, in this membrane, we have phospholipids, but we also have these proteins that can insert through the membrane. They can sometimes be attached to just one side of the membrane, and this can be a place where proteins or molecules go into and out of the cell, or they can be places where the cell has a receptor or interacts with the outside environment. We can also sometimes see these sugars attached to different proteins or lipids on the outside surface of the cell. Here is an example of a transmembrane protein. Now, look at this red alpha helices in this protein. You see it's all, the secondary structure of this protein contains all alpha helices, and it's usually conformed in a way that these are all hydrophobic amino acids. And that's because the phospholipid bilayer is made out of lipids. So we talked before about lipids, how they have fatty acids, and in a phospholipid we have two fatty acid chains. So we can see that here in these green and white. We know that these are non-polar hydrophobic molecules. They don't want anything to do with water. In this particular molecule, instead of a third fatty acid that would make fat, we add a phosphate group. Now remember, our phosphate groups have those negative charges, so they are polar, they love to interact with water, they are hydrophilic. So we have this amphipathic molecule where we have a polar region and a non-polar region. Now if you just drop these types of molecules into water, which is of course a polar molecule, they will self-assemble so that all these yellow non-polar regions are in the inside, sort of hidden away, and only the polar regions, these phosphate groups, are exposed to the outside. So we see, right, that we have this phospholipid bilayer. We have two layers of phospholipids with their hydrophobic tails on the inside. We call this sort of the outer leaflet, these phospholipids, and the inside phospholipids are the inner leaflet. So this whole region here is nonpolar, and that's oftentimes why we have to have protein channels to help move things through the membrane, and why those nonpolar proteins end up in the middle here. So we have this polar aqueous extracellular environment, we have this polar aqueous cytoplasmic environment, and in between we have this nonpolar barrier. So this is an example of how the structure, right, with both nonpolar and polar regions, helps it make a really effective barrier with a very watery world, right, both inside the cell and outside of the cell. Okay, so how do things move through the membranes? We talked a little bit about those proteins that can help do it, but some molecules can travel through the membrane by themselves and through a process of diffusion. So there's hydrophobic molecules that are small, and these are freely permeable. They can sort of wiggle in between those phospholipid bilayers and move. Some of those are oxygen, carbon dioxide, and nitrogen gases can do this. Now, some molecules are small enough and they're uncharged, so they can still sort of pass through. So water and glycerol can move and are considered somewhat permeable, right? Now, there are some things that cannot move through the membranes without assisted help. These would be the kinds of molecules that we need to have protein channels for to help us move them from one place to the next. These are large molecules, uncharged molecules, things like glucose and sucrose. So all those sugars need to be able to move it through the cell. They have to do it through different protein channels. And then those ions, so because they're really polar, they can't make it through that non-polar region in the middle here. So we consider these to be impermeable as well. So we have our ion channels for all of our different important ions. Okay, so I've already alluded to the fact that there are things that move through the cell. Let's back up a second. Through diffusion. So diffusion is the movement of molecules from a high concentration to a low concentration. So everything wants to sort of equal out and balance. So if there's too much of a molecule on one side of the membrane, it will move the most molecules to the outside to sort of help balance that out. And depending on the molecule, it may do that easily or not so easily and require protein channels. So we can have facilitated diffusion and even active transport to help move the molecules across. The other way that things can move is through osmosis. Now, osmosis, instead of moving the molecules, we move the water. So this is the movement of water from an area that's less concentrated to an area that's higher concentrated. So when the solutes, or the particles in the solution, are balanced, meaning there's the same number of salts or solutes inside the cell and outside the cell, we call it an isotonic solution. And nothing really has to change. Everything seems to be in balance. So the movement of water across the membrane has no effect. Nothing really happens. Everything's sort of balanced out. Now, if the cell finds itself in an environment where it's higher concentration outside of the cell, so a salty environment or a high sugar concentration outside of the cell, we call that a hypertonic solution. And water will leave the cell to help balance that concentration out. So here you see the cell shrinks actually because the water has left, and we now have a more balanced concentration gradient. So we call this osmotic osmolysis, right? So those are the cells sort of shrinks down, plasmolysis, so it shrinks down and shrivels up. So I always think about grapes, right? So a raisin is not a happy grape. So cells can't really live and survive like this for very long. So oftentimes they will die because all the water has left the cell. Now the opposite thing can happen as well. In a hypotonic solution, where there's more solute inside the cell and less outside, water will rush in. And if too much water enters, the cell can lyse. And so we call this osmotic lysis. All right. So the last thing we want to talk about with membranes is how do things move into the cell. So we talk about two different processes, endocytosis and phagocytosis. And they're different basically on the size of particles, and there's some overlap. So endocytosis we mostly think about as how viruses are very small get into the cell, particles can get in the cell, and even some small bacteria will get into the cell through endocytosis. Larger things will enter the cell through phagocytosis, which is a more facilitated process. During the immune system, the cell can bind to, with antibodies, can bind to things and bring them in through phagocytosis. So this is sort of a bigger way of eating larger things. Now, when we think about this process, we have something binding the surface and then bringing it in into having to this internal membrane structure. So here we see endocytosis occurring. These little blue particles are getting endocytosed, and this is called an endosome. Now, many different kinds of cells have these internal membranes or these internal membrane compartments. We might have an endosome, but we might also have other compartments within the cell that are divided by a membrane compartment. And those are called organelles or intermembrane organization. And the internal membrane compartments are really only found in eukaryotic cells because they're so large. Prokaryotic cells have one single membrane, plasma membrane, that leads from the inside of the cell to the outside of the cell. They have no internal membrane compartments to divide things up. So when we see this endocytosis and phagocytosis, we should be remembering that these are happening within eukaryotic cells. Okay, so here is an EM picture. So again, the electron microscope allows us to see this. So here's the membrane of the eukaryotic cell, and it's engulfing these particles here. So this endosome is being formed. Okay, now when a cell needs to eat something larger, we said it's going to do that through phagocytosis. So here we have an amoeba, this big old amoeba, and it's reaching out and it's going to eat this paramecium. Typically, phagocytosis involves receptor binding, it involves sort of attaching to whatever it's going to eat, and then pseudopods, or these sort of arms, go around and give the organism this sort of engulfing hug, and it gets brought into the phagosome. So it's, again, used for larger molecules or eating. We see this paramecium that the amoeba is trying to eat, but it's also already had one for lunch here too. Interestingly enough, the paramecium used endocytosis to eat bacteria, and this amoeba is using phagocytosis to eat the paramecium. So we're kind of looking at a food chain here. And notice our scale bar, we can kind of get an idea of how big this is. Okay, so our last case study is at the end of this chapter, and here we have a 28-year-old girl, and she has overnight developed severe cramps, stomach cramps, and diarrhea. She spikes a pretty high fever and thinks, oh, maybe I just have a stomach bug. She kind of waits it out, but after 3 days, she's still having horrible diarrhea, and blood is now appearing in her stool. So she goes to the emergency room. In the emergency room, they begin treating her with antibiotics and running some tests to find out what she might have. And it turns out she is positive for Salmonella enterica Serovar Heidelberg, right? So this is a foodborne pathogen. They think she probably acquired it from some improperly handled chicken, which is why we have this chicken guy standing here. And this particular kind of bacteria is really good at busting out and preventing digestion after endocytosis. And that's why we're talking about it here in this part of the chapter. So here's an electron microscope of a intestinal cell that has been infected by Salmonella. So the Salmonella bacteria get endocytosed, and instead of fusing with the digestive compartment called a lysosome, these bacteria prevent that from happening, and instead don't die during endocytosis. So they get inside the cell and they survive inside the cell, and the cell becomes sort of their safe background sort of environment that they can operate from. So when this particular girl was treated with antibiotics, It was not very successful because the antibiotics had a hard time penetrating the host cell and then getting to the bacteria that were inside of that cell. So oftentimes these infections are harder to treat because the Salmonella bacteria is hiding out inside the cell after busting out of the endosome. Another great bacteria that does this, that's really good at evading sort of getting eaten, is called Listeria. All right, so that ends our discussion in Chapter 4, really thinking about the chemistry of the different parts of cells and why we need to understand how the different organic molecules work, and giving us a nice introduction into membranes and how molecules